Now, how do we image a single molecule? So first of all, we're pumping electronic transitions, a ground to excited state, electronic transition. Now we're going to collect the emitted fluorescence terminating on vibrational levels of the ground state. And if we do that, we're going to be able to see each single molecule as a dot. And so think of it this way. A laser is sent through a microscope. It's focused with an objective, which I cannot point to. Then the emitted light is shifted to long wavelengths due to the Stokes shift. And so the detection system, which is a camera or so forth, needs filters to block out the pumping laser light in order to detect the light from the molecule. And it's worth noting right here that even your own eye, you can look into my microscope and you can see the light from these single molecules. Your eyes are sensitive enough to be able to detect the light from individual molecules. Of course, we have to have the right laser color, and we have to collect well, and we have to do various things to make this work easily, but it's done now all over the world in many laboratories, including, of course, in Russia. So, thinking about this from a slightly different way, let's look here at the picture on the right, and you see most often, maybe you want to study a protein that's not fluorescent. Then what we will do is you'll attach a small fluorescent dye to that molecule, and then it's going to be the light from that fluorescent label that you're going to be detecting. So these labels are rhodamine dyes, cyanine dyes, other good emissive dyes, fluorescent proteins like green fluorescent protein, even antenna proteins from photosynthesis. But now imagine you're focusing your laser to a small spot, and remember that diffraction effect that I mentioned a few moments ago. It limits the minimum size of that spot to a few hundred nanometers. So the molecules are 1 nanometer in size, but this laser spot is a few hundred nanometers in size. So now you see that to pump a single molecule, you have to dilute them. You have to make sure that they're farther apart than the size of this laser beam. So if you do that, you'll be able to detect single molecules done carefully. And here's an example, hoping that this important video, yes. So this is now in a cell, and I'm looking at a transmembrane protein that's important in your immune system. It's been labeled, and you can see at room temperature, there's a spectacular dance of these molecules on the surface of the cell. Much information is hidden in that motion. How important is cholesterol? What is the diffusion coefficient? Is it normal motion or not? And let's look at it again, because this little movie, which is some kind of old, it's still really beautiful to me, because not only do you see the motion, you also see some of the molecules disappear. What does that mean? You notice in one step they disappear. That's a proof that it's a single molecule, but this is photobleaching, okay, on the single molecule level. You don't see an exponential decay. You see the molecule simply disappear, and that's some proof that it is an individual molecule. So there's other things in that movie, but because of time, I'll just keep going. So now that people started studying single molecules at room temperature in the mid-1990s, surprises started to appear. Again, just like at low temperatures. So what surprises? On the left are bacterial cells in white light. So I'm just showing you in white light a few bacterial cells. They're pretty small. But on the right, I'm showing you the light coming from that sample. And in these bacteria, a particular protein has been fused to yellow fluorescent protein. So this fluorescent protein is a label, and you're looking at the light from the label, right? What happens, though, if I just run the movie and show you what happens? It's amazing. These molecules are blinking. This was the surprise. We didn't expect this for fluorescent proteins at room temperature. So each one of these flashes of light are coming from inside of those bacterial cells. And the movie contains a great deal of information about all of those individual single molecules. And here there's no motion. So please don't be confused. That earlier movie was showing motion moving around. Here there's no motion. All of this is just the blinking of fixed molecules, which are going to be useful in a few minutes. But you see, again, people said, oh, what good are single molecules? They blink too much and so on. It's not so interesting. Well, we'll see. But when you explore a new regime, it's a wonderful aspect that you really can't predict in advance some of the surprises that might occur if you hadn't done the experiments. This is why basic science is so essential. Doing the experiments, you can find out if there are surprises. And the surprises can lead to some unexpected and unplanned applications. So I'll tell you about that. Now let's start talking about the bigger problem, okay? The big problem here is we'd like to observe in these cells, in this case a bacterium, for example, all of these nanoscale machines that are at work inside. I mean, you can see DNA and proteins and so on. Looking more closely, you can see motors, like the motors that were mentioned in the Professor Savage's talk, and you'd like to be able to see what they're doing to learn about this interesting system. So you want to have a technique that has high sensitivity and high specificity, and possibly living cells, and high spatial resolution. Because look at the scale bar here. You see that little 20 nanometer. These objects are really tiny. So you might say, well, you could use electron microscopy, but in electron microscopy, all of this just becomes a fuzzy blur of gray objects, gray and white objects. So you want some specificity, and that's what fluorescence does for you. It lets you label a specific set of molecules so that the light only comes from them. However, here's the problem. The smallest possible visible laser spot is too big compared to the size of all these objects. The diffraction limit, again, is limiting it. So that's what led to super-resolution microscopy, and Stefan Hell used a particular technique called stimulated emission depletion. Eric Betzig and I used single molecules in the way I'm going to describe now. So once again, explaining what we're talking about. Here's the bacterial cell again, and we've labeled now a particular protein with these fluorescent proteins, and you're seeing the light and you want to see the structure. You might think all you have to do is to buy the most expensive microscope possible. So here it is. Here's this most expensive microscope, and you don't see anything. That's because each of the individuals, even though they're only a few nanometers in size, they appear to be a few hundred nanometers in size due to that Abbe's diffraction limit. So when you use super resolution, however, this image becomes that image. And that's the difference. Before, you could not possibly see the detail, and there was no way to make it better because it was fundamentally blurry due to the diffraction limit. But with super resolution, we can remove that limitation and see this detail down to the 40 nanometer scale in this case. So how does it work? The first idea is a simple one: super localized single molecules. Think of this little mountain, which is in a lake in Oregon. And you know that if you just walk up to the top of the lake with your cell phone, you can just simply read out the coordinates of the top of the mountain. And of course, I have to have a scale bar there. It's 120 times 10 to the 9th nanometers, okay?